Tinea cruris is a superficial fungal infection caused by dermatophytosis that have the ability to adhere to keratin and use it as a source of nutrition. By attacking keratinated system such as the stratum corneum in the epidermis, hair, and nails. Tinea cruris is caused by a fungus of the genus of Trichopiton sp that usually live in humid place. This is supported by the situation in Indonesia, where Indonesia is a tropical country and has a humid climate. This condition makes it easy for fungi to grow and infect human skin. Currently, the treatment of Tinea cruris using synthetic drugs are available in store drugs used both oral and topical drugs in adult class. However, the use of synthetic drugs has side effects such as rash, itching, diarrhea, and abdominal pain, which will cause you problems for patients. In addition, the use of topical drugs will increase fungal resistance to drugs. So, synthetic drugs have side effects that must be stopped immediately and are now starting to switch to drugs that contain natural ingredients. Therefore, Hafiez Arioso, proudly present my innovation is aloe vera and Citrus alfantiora spray. This spray is a spray made from a combination of aloe vera and Citrus alfantiora extract. Substances that act as antifungal are aloe vera gel, which has active compounds of saponins, anthraquinones, flavonoids, polyphenol, and phenol, which are effective for antifungal use. Compounds that affect the performance of fungal cells so that fungal cells cannot work normally. Eventually, the cells will die. On the one main content pill of Citrus alfantiora is containing flavonoids, saponins, and essential oils. Based on research conducted by her 2011, the pill of Citrus alfantiora has the ability to inhibit the growth of fungi cascadiaceae such as Trichopiton metragophytus fungi. It is hoped that this combination can increase antifungal compounds in inhibiting the growth of the Trichopiton sp that causes Tinea cruris. The novelty in this research is in the antifungal material used. In previous research showed no research has been conducted using a combination of aloe vera and Citrus alfantiora in inhibiting the growth of the Trichopiton sp that causes Tinea cruris. The work step in this research is the first step is scraping a skin sample by a doctor specialist skin and genital at Blambangan Hospital Banyuwangi. This research has received ethical approval from the Health Research Ethics Ethicus Banyuwangi. The second step is making of potato dextrose agar, keratin agar, and skim medium agar for the keratin and protease degradation test. Keratin is obtained by extracting it from chicken's feathers. The third step is the maceration extraction process. Aloe vera and peel of Citrus alfantiora extract by maceration using 96% ethanol. The first step is to carry out phytochemical tests on the extracts to test the content of flavonoid, tannins, saponins, and essential oil. The fifth step is to plant the mushroom sample on the medium. The sixth step is an antifungal effectiveness test. Next, the results and discussion. In macroscopic view, Trichopiton shows the growth of the mycelium or hive. The smooth one is white and look like cotton, although sometimes it can be other colors depending on the interesting pigment. In the microscopic view, Trichopiton has hive or mycelium which generally not insulate, except in the part of the hive that will form or produce conidia. Birds of hive are generally short with a relative thin shape. Is the result of hive-bud bonding. Hypospiral mitochondria around macroconidia are pencil-shaped and consist of several cells. In the keratin degradation test, from the picture, you can see a clear zone around the mushroom. This signifies that the fungus degrades the surrounding keratin. In addition, the growth of fungi is relatively more faster than in the non-keratinized medium. This is due to fungi is nourished by the presence of keratin, which is a nutrient for fungi. Dermatophytosis is a superficial fungal infection caused by the dermatophytosis that has the ability to adhere to keratin and use it as source of nutrition. By attacking keratination tissues such as the stratum corneum in epidermis, hair, and nail, Verma 2008. In Verma research, strengthened suspicion that the mushroom plant are the dermatophytosis using keratin as source of nutrition is characterized by the presence of a clear zone and variety of mushroom growing using a keratinized medium. In the proteolytic or protein enzyme test is an enzyme test of fungi that have the ability to decompose or break down protein molecules. Colonies of proteolytic fungi can be recognized by the presence of a clear area around the colony, which indicates casein hydrolase. In this research, phytochemical tests were carried out to determine the content of phytochemical from extract of aloe vera and peel of Citrus althanthera. Phytochemical testing on this research uses a qualitative method where the reaction that occurs in the extract after being given a reagent, the color and shape of the extract liquid will be observed tested. The compounds tested in this research were flavonoids, saponins, tannin, and essential oil. Based on the result of the phytochemical test, it was found that the flavonoid test had positive results are indicated by the formation of a yellowish-green color. The saponin test was positive, indicated by the formation of a stable foam for 10 minutes after the extract was given distilled water and shaken for 30 seconds. The tannin test has a positive result indicated by a color change to blue-blackish after the extract was reacted with 1% FeCl3. Test essential oil have positive results after being reacted by dissolving 1 ml of the extract was passed over the cup until the residue was obtained. Positive result of essential oil characterized by a distinctive odor produced by the research. In the picture, it appears around the paper disc on each there is a reaction in the test solution of aloe vera, peel of Citrus althanthera, the zone overgrown with fungus in fading. When compared, the positive control have in common that there is fading of the colony around the paper disc. The fading around the paper disc is marked with difference in color and density of fungal mycelium, different from negative control that did not have any reaction around the disc. Fading the fungus occurs because the fungus that grows interacts directly with the antifungal compounds from extract and positive control. After transfers cut were made and observed from above, the image was observed for fungal growth under the paper disc. Observation result in the form of no fungal growth under the extract solution or positive control. In contrast to the negative control figure, on the negative control still had fungal growth. This happened because negative control in the form of distilled water did not have antifungal properties and caused mushroom growth. Next, the conclusion. Based on the result of the research, it can be concluded that administration of aloe vera and peel of Citrus althanthera extract affects Trichobiton rubrum, Trichobiton sp fungus. Growth is characterized by the absence of fungus growth Trichobiton sp under the disc treated with aloe vera extract and peel of Citrus althanthera extract at a concentration of 100%.